The next day, both Wilkins and Franklin came up to see what we had done. The threat of unanticipated competition briefly united them in common purpose. Franklin wasted no time in faulting our basic concept. My memory was that she reported almost no water present in crystalline DNA. In fact, the opposite was true. Being a crystallographic novice, I had confused the terms unit cell and asymmetric unit. Crystalline DNA was in fact water-rich. Consequently, Franklin pointed out, the backbone had to be on the outside and not, as we had it, in the center, if only to accommodate all the water molecules she had observed in her crystals. That unfortunate November day cast a very long shadow. Franklin's opposition to model building was reinforced. Doing experiments, not playing with tinker-toy representations of atoms, was the way she intended to proceed. Even worse, Sir Lawrence Bragg passed down the word that Crick and I should desist from all further attempts at building a DNA model. It was further decreed that DNA research should be left to the King's Lab, with Cambridge continuing to focus solely on proteins. There was no sense in two MRC-funded labs competing against each other. With no more bright ideas up our sleeves, Crick and I were reluctantly forced to back off, at least for the time being. It was not a good moment to be condemned to the DNA sidelines. Linus Pauling had written Wilkins to request a copy of the crystalline DNA diffraction pattern. Though Wilkins had declined, saying he wanted more time to interpret it himself, Pauling was hardly obliged to depend on data from King's. If he wished, he could easily start serious X-ray diffraction studies at Caltech. The following spring, I duly turned away from DNA and set about extending pre-war studies on the pencil-shaped tobacco mosaic virus using the Cavendish's powerful new X-ray beam. This light, experimental workload gave me plenty of time to wander through various Cambridge libraries. In the zoology building, I read Erwin Kargaff's paper describing his findings that the DNA bases adenine and thymine occurred in roughly equal amounts, as did the bases guanine and cytosine. Hearing of these 1-to-1 ratios, Crick wondered whether during DNA duplication, adenine residues might be attracted to thymine, and vice versa, and whether a corresponding attraction might exist between guanine and cytosine. If so, base sequences on the parental chains, for example ATGC, would have to be complementary to those on daughter strands, yielding in this case TACG. These remained idle thoughts until Erwin Kargaff came through Cambridge in the summer of 1952, on his way to the International Biochemical Conference in Paris. Kargaff expressed annoyance that neither Crick nor I saw the need to know the chemical structures of the 4 bases. He was even more upset when we told him that we could simply look up the structures in textbooks as the need arose. I was left hoping that Kargaff's data would prove irrelevant. Crick, however, was energized to do several experiments looking for molecular sandwiches that might form when adenine and thymine, or alternatively guanine and cytosine, were mixed together in solution. But his experiments went nowhere. Like Kargaff, Linus Pauling was attending the International Biochemical Congress, where the big news was the latest result from the Phage group. Alfred Hershey and Martha Chase at Cold Spring Harbor had just confirmed Avery's transforming principle. DNA was the hereditary material. Hershey and Chase proved that only the DNA of the Phage virus enters bacterial cells. Its protein coat remains on the outside. It was more obvious than ever that DNA must be understood at the molecular level if we were to uncover the essence of the gene. With Hershey and Chase's result the talk of the town, I was sure that Pauling would now bring his formidable intellect and chemical wisdom to bear on the problem of DNA. Early in 1953, Pauling did indeed publish a paper outlining the structure of DNA. Reading it anxiously, I saw that he was proposing a 3-chain model with sugar phosphate backbones forming a dense central core. Superficially, it was similar to our botched model of 15 months earlier. But instead of using positively charged atoms, for example magnesium 2+, to stabilize the negatively charged backbones, Pauling made the unorthodox suggestion that the phosphates were held together by hydrogen bonds. But it seemed to me, the biologist, that such hydrogen bonds required extremely acidic conditions never found in cells. With a mad dash to Alexander Todd's nearby organic chemistry lab, my belief was confirmed. The impossible had happened. The world's best-known, if not best, chemist had gotten his chemistry wrong. In effect, Pauling had knocked the A off of DNA. Our quarry was deoxyribonucleic acid, but the structure he was proposing was not even acidic. Hurriedly, I took the manuscript to London to inform Wilkins and Franklin they were still in the game. Convinced that DNA was not a helix, Franklin had no wish even to read the article and deal with the distraction of Pauling's helical ideas, even when I offered Crick's arguments for helices. Wilkins, however, was very interested indeed in the news I brought. He was now more certain than ever that DNA was helical. To prove the point, he showed me a photograph obtained more than 6 months earlier by Franklin's graduate student, Raymond Gosling, who had X-rayed the so-called B form of DNA. Until that moment, I didn't know a B form even existed. Franklin had put this picture aside, preferring to concentrate on the A form, which he thought would more likely yield useful data. The X-ray pattern of this B form was a distinct cross. Since Crick and others had already deduced that such a pattern of reflections would be created by a helix, this evidence made it clear that DNA had to be a helix. In fact, despite Franklin's reservations, this was no surprise. Geometry itself suggested that a helix was the most logical arrangement for a long string of repeating units, such as the nucleotides of DNA. But we still did not know what that helix looked like, nor how many chains it contained. The time had come to resume building helical models of DNA. Pauling was bound to realize soon enough that his brainchild was wrong. I urged Wilkins to waste no time, but he wanted to wait until Franklin had completed her scheduled departure for another lab later that spring. She decided to move on to avoid the unpleasantness at King's. Before leaving, she had been ordered to stop further work with DNA, and had already passed on many of her diffraction images to Wilkins.